First off, thank you, everyone. Thank you to everyone for showing up today. If you tuned in early, heard Nate Zabota and I talking about the lovely weather up here in Alaska. I saw fresh snow on the peaks near me yesterday at only about 6,000 feet, and it's a high of 52 today. So if you're someplace hot, love to maybe do a swap. Today, I'd like to invite or introduce Jack Hopkins, who is the president and CEO and assistant professor and all sorts of things at the Center for Wildlife Studies. Unfortunately, Jack could not join us today. I have a recording. Please stay with us. He made a recording of his presentation. So it was a very much last minute event that happened last night. In fact, he wasn't even sure as of this morning what was going to happen, but he's able to record his presentation for us. And I can honestly and objectively say that this is some very exciting stuff that he's working on. I've always been a bit of a skeptic when it comes to stabilized analyses. I've been involved with some of it in the past. But I know there's also a very, it's much more complicated than it may seem on the surface. But what Jack has been working on is truly exciting stuff. There's a good chance that he'll be able to join us for the Q&A, which is another reason to stick around. If by any chance he can't, then I will definitely encourage people to reach out to him directly. And as always, we will post this video to our YouTube playlist in a day or so. But again, feel free to reach out, as Jack will say here in a moment as a recording. We'd love to hear from people. And this series is pretty important to us. It's a good chance for us to highlight the work of our collaborators and instructors, to promote our courses and our research. It's really been one of the highlights of my time here with the center.
Hi, everyone. Thanks for showing up for my talk today, especially since I couldn't make it. Maybe I'll see you at the Q&A at the end, but please don't hold your breath. Before I begin, I've got to make a shameless plug because I'm the president about our organization, Center for Wildlife Studies. Since 2020, in COVID, we've been providing specialized training to folks like you all over the world in the quantitative sciences, GIS, conservation planning, field methods, on and on. Our instructors are experts from all over the world, and many are also active researchers who are excited to share their work in forums like this, like Wildlife Wednesday, or bring it into their classrooms. After the talk, please have a look around our website, consider supporting our organization by taking a course, have a look at our scholarship program, and we're a 501c3 nonprofit, so any donations that you make are tax deductible. Either way, thank you so much for being here. I really appreciate the support of our organization and myself. Please feel free to reach out to myself or any of our staff. We'd be happy to answer your questions. Thanks. Take care. Enjoy the talk.
Okay, so today I'm going to talk about some of my new preliminary work using laser ablation to study black bear foraging ecology. Before I begin, I'd like to thank Jim Moran at Michigan State, my good buddy and long-term collaborator Jake Ferguson at the University of Kentucky, and graduate students Yang Wang and Katie Rock. Before I introduce how laser ablation mass spectrometry is helping us reconstruct the fine-scale diets of bears through time, I'm going to cover a few preliminary topics that led me to this cool new technique. First, I need to talk a little bit about American black bears, the main focal species of this talk, and their foraging behavior. Because if for no other reason, like maybe some of you, I'm a huge black bear geek and have been for some time. Next, I'll provide a general introduction about how stable isotope analysis is used to study the diets of animals, leaning on some of my past work with bears as case studies. So let's begin with the American black bear species classification as an organism, which will shed light on understanding their diet and foraging behavior. Since this is not a full-on lecture about bear evolution, I'll skip the details about early levels of classification and start with introducing the 8 bear species of the world: the American black bear, brown bear, polar bear, Asiatic black bear, sun bear, sloth bear, spectacled bear or Andean bear, and giant panda, as well as their membership in the class Mammalia, which are animals characterized as having hair, mammary glands, and warm-blooded physiology, just to name a few important ones. And then on to carnivora, the order that includes bears and other mammals such as wolves, cats, seals, and related taxa. Like bears, other members of this order have forward-facing eyes, clawed feet, and have monogastric digestive tracts, just like us, which are relatively simple compared to herbivores' multi-chambered stomachs. Bears are also members of the family Ursidae, the bear family. Their large, heavy body build, plantigrade stance, that's walking flat-foot like humans, 5 toes with strong non-retractable claws, short tails, and a more generalized skull and tooth structure suited for an omnivorous diet separates them from other carnivores such as canids, felids, and mustelids. Within Ursidae, there are 4 species of true bears, including the American black bear, which is a member of the genus Ursus that includes 3 other evolutionary-related large-bodied bears with a generalized omnivorous anatomy, distinguishing them from more specialized bear lineages such as pandas, sloth bears, spectacled bears, and sun bears. Interestingly, polar bears are also included in the genus Ursus because they evolved from within the brown bear, true bear lineage and retain the core anatomical traits of Ursus, even though they are now highly specialized for a carnivorous marine lifestyle. And then finally, we have Ursus americanus, the American black bear. Black bears originated on a separate evolutionary lineage from other Ursus bears with distinct genetics, geography, body form, ecology, and behavior. They are derived from a forest-adapted lineage that occupies North American forests, mountains, wetlands, and human-dominated landscapes across much of the continent, whereas brown bears became larger terrestrial omnivores and polar bears became specialized Arctic marine mammals. Compared to brown bears and polar bears, American black bears are generally smaller and don't have a hump on their shoulders, like grizzlies. Grizzly bears typically have a more pronounced dished or concave facial profile, and black bears have bigger ears. Black bears have shorter curved claws adapted for climbing and a highly flexible omnivorous diet. Black bears forage on a wide range of foods, including green vegetation, fruits, hard mass such as acorns, carrion, live prey and insects, and in some places human foods. What an individual bear eats can vary dramatically depending on where it lives, the season, its age and sex, and the opportunities available in the landscape. This flexibility is one reason black bears are so successful as a species, but it also makes their diets difficult to measure. Understanding what bears eat is important because foraging choices can influence human-bear conflict, survival, reproduction, population dynamics, and the management actions we use to conserve bear populations and reduce conflicts with people. Some black bears are often in conflict with people and their property because they also forage for anthropogenic foods, or human-derived foods, that are calorie-rich and consistently available to bears where people live. When bears learn where to find these foods, they often become a big management concern because these bears become habituated. These are bears that show little to no overt reaction. They just don't visibly respond to people as a result of being repeatedly exposed to the anthropogenic stimulus that they're presented with without substantial consequence. These habituated bears often become human food conditioned. A food conditioned bear is a bear that has learned to associate people, human activities, human use areas, or food storage receptacles with human-derived food. These bears are the ones that we often hear about and see and are often the focus of most human bear management programs. For management purposes, bear incidents and sightings are typically recorded and tracked by park staff and state fish and game agencies. Because there's often differences in reporting across jurisdictions, in 2010, we asked 25 professionals engaged in human bear management and research in the United States and Canada to review the current terms and concepts currently used in human bear management and research, then work together to arrive at a consensus of definitions among co-authors. First, here we made the distinction that an incident is not a bear sighting. A sighting occurs outside a bear's ORD, or overt reaction distance, which is the distance at which a bear visibly responds to people during a human-bear interaction. On the other hand, bear incidents occur inside a bear's ORD, when a bear exhibited stress-related or curious behavior and a person took extreme evasive action, or when a person or bear was injured or killed. These human-bear conflicts are recorded as bear incidents, as well as instances which may not involve human-bear interactions. Instead, there is no conflict with people, but these bears cause property damage, commandeer anthropogenic foods, injure people's pets, or were involved in vehicle collisions. Bear incident reports record data on the interactions bears have with people and/or their property, including human food. Although these reports do a good job recording incidents about tagged and monitored bears that were observed by staff or visitors, they sure don't do very well at recording information about the incidents these bears were involved in when unobserved. And these reports are often not linked to bears because they are not monitored by management staff at all. 20 years ago, I was hired by Yosemite National Park as a graduate student to determine the severity of the human bear problem in the park. That's outside of what park staff had documented about known bears involved in bear incidents. So how did I do it? I relied heavily on the analysis of stable isotope ratios and my good buddy and mentor, Paul Kosh, professor of Earth and Planetary Sciences from the University of California, Santa Cruz. Okay, I'm going to come back to this later. So before I get into all that, I need to talk about stable isotopes and their use in ecology. As a refresher, isotopes are atoms with the same number of protons, but a different number of neutrons, resulting in each variant having a different atomic weight. Isotopes range from 2 to 36 per element, whereas stable isotopes range from 1 to 10. The stable ones are the ones we're interested in because they're not going anywhere. As a result, we can use them as chemical tracers. Stable isotope analysis uses an elemental analyzer and an isotope ratio mass spectrometer as an automated team. The EA prepares and combusts the sample, and the IRMS separates and measures the relative abundance of a heavier isotope to a lighter isotope of the same element in a sample. This ratio in the numerator is then compared to an international standard in the denominator. And the small differences between the two make it easier to measure, compare, and interpret the resulting delta values, which are in parts per thousand or per mil. A positive value means the sample is heavier than the standard, whereas a negative value means it's lighter or depleted in heavy isotope when compared to the sample. Stable isotopes are used in lots of ways in ecology. Today I'm going to talk a little bit about stable isotopes as chemical tracers to understand the structure of food webs. They're also used to detect animals that are specialists or by default generalists, like bears that commandeer human food versus those that are just natural foragers. And the last application I'll talk about today is estimating the diets of animals based on the isotopic composition of their foods and their tissues. To begin, let's first learn a bit about resolving the trophic structure of food webs using stable isotopes. To do that, let's focus on carbon stable isotopes first, since all known biological life forms are carbon-based. Carbon comes in slightly different forms, including a lighter form and a heavier form. Plants take up carbon from the air in both forms and use it to build their tissues. C3 plants, which include most trees, shrubs, berries, and cool season plants, are better at using the lighter form of carbon, so their tissues end up with less of the heavier form. C4 plants, on the other hand, such as corn and sugarcane, use a different photosynthetic pathway that does not sort the lighter and heavier carbon as strongly. Because of that, corn and sugarcane have a different carbon signature than most natural foods in many forest ecosystems in that they're heavier. So as a rule of thumb for delta C13, you are what you eat, except that each consumer's tissue, such as this deer that you see here, are predictably a little heavier than its foods because metabolism fractionates or sorts isotopes, and nutrients are routed differently into tissues, each tissue recording diet over a different window of time. Plants also carry a nitrogen signature, but unlike carbon isotopes, the difference is usually not caused by the plant's photosynthetic pathway. Instead, nitrogen isotopes often reflect where the plant got its nitrogen and what happened to that nitrogen in the soil before the plant absorbed it. Nitrogen comes in a lighter form and a heavier form as well. Soil microbes, fertilizers, animal waste, decomposing plants, and nitrogen-fixing bacteria can all change the balance of light and heavy nitrogen available to plants. As a result, plants grow in different soils, using different nitrogen sources, or growing in areas influenced by animal waste or human activity can have different nitrogen isotope signatures. Nitrogen isotopes help indicate where an animal feeds in the food chain or web. As animals process protein, they tend to lose slightly more of the lighter nitrogen isotope in waste, especially urine, leaving their tissues with relatively more of the heavier nitrogen isotope. This is why nitrogen isotope values generally increase from plants to herbivores to carnivores in predictable 3 to 4 per mil steps at each trophic level. Interestingly, black bears in Yosemite that have natural diets of plants, berries, and nuts look a lot like deer isotopically, except for a little bit different C3 diet when compared to deer, indicating sampled bears were primarily vegetarians in this system. As for other bear species, such as coastal brown bears in Alaska, they look quite a bit different isotopically. Here you can see they're about 10 per mil higher in delta N15 because they feed on Pacific salmon that are at the 4th trophic level. Salmon are a large predatory fish that feed on smaller fish that feed on zooplankton, copepods, krill, and other invertebrates, which then feed on phytoplankton and algae, which are primary producers. So what do you think about people and Americans in specific? Where do you think they land in this plot?
People occupy anywhere between the 2nd and 5th trophic level, depending on where they live in the world and what food habits they have. But Americans are at the higher end of that range because we eat a lot of beef and fish, and we're also heavy in corn. No pun intended. Not because we eat a ton of corn directly,
but because we eat processed foods high in high fructose corn syrup. Understanding the isotopic relationship between consumers and their food sources, as well as the isotopic differences between conspecifics that are known to forage differently, allows us to then detect animals that are specialized from a sample of individuals with unknown foraging behaviors. To demonstrate this, here's some of my past work where we measured carbon and nitrogen isotope values in black bear guard hair. This was a whole hair analysis, collected from bears throughout Yosemite National Park. Each point here represents a bear hair sample, the full hair. Black circles are known food-conditioned bears that were radio-collared and monitored. The black squares are known non-food conditioned bears. These bears were also fitted with radio collars at the same time and were confirmed not to be food conditioned. And lastly, we have gray circles, which are unknown bears sampled throughout the park using hair snares. We learned that bears known to consume human foods generally had higher nitrogen isotope values than bears that were not known to consume human foods, and delta C13 alone was not very helpful for discriminating between food-conditioned and non-food conditioned bears in Yosemite. Because delta N15 was higher in food-conditioned bears, we used delta N15 isotope values as a predictor in a logistic regression model to learn that an additional 19 bears that nobody knew about or likely human food condition. The importance of this finding was that it demonstrated the use of stable isotopes in hair as a non-invasive way to identify bears foraging for human food in locations that the park staff didn't know about. Another project where we used stable isotope analysis to detect dietary specialization was in Banff National Park. There, we studied the impact of the Canadian Pacific Railway on protected grizzly bears in the park. Grizzly bears that use the railway in Banff forage on plants and a variety of human-derived foods, including train-killed animals and spilled grains being carried by the rail, including wheat, barley, like you see in this photo, canola, lentils, peas, and corn. Here we compared 3 stable isotope systems: carbon, nitrogen, and sulfur, derived from the hair of grizzly bears that were killed on or captured near the railway, with bears sampled away from the railway using hair snares. Similar to food condition bears in Yosemite, we found that the rail-associated bears had higher nitrogen isotopes than hair-snared bears. Higher delta N15 suggests that rail-associated bears consumed more animal protein, likely from scavenging animals killed by trains. We also found that higher delta S34 values in rail-associated bears, when compared to hair-snared bears, likely reflected the direct feeding of sulfur pellets spilled along the tracks or indirect exposure of sulfur associated with the rail corridor in general, including sulfur taken up by plants and animals near the railway. Lastly, we found that delta C13 values were similar between rail-associated bears and hair-snared bears, suggesting that the two groups had similar plant-based diets, and that spilled corn on the railway, which there really wasn't much of, unfortunately, or other C4-derived foods were not an important enough food source for bears in our study to separate the groups isotopically. Overall, we learned that nitrogen and sulfur isotopes in hair are greater than conspecifics, which can help identify bears that use the railway, those are the bears in black, and provide a non-invasive tool for detecting individuals at greater risk of train-caused mortality and for guiding mitigation efforts on the rail. Okay, now I'd like to transition into something I focused a lot of time on, which is estimating the proportion of dietary contributions to animals' diets using stable isotopes. Unlike scat or gut content analysis, this analysis does not directly measure the frequency or volume of food in an animal's diet or the calories based on what is eliminated from an animal or found in its guts, but instead provides estimates of the foods assimilated into their tissues. Here's how this works in a nutshell. No pun intended. Here I'll use 2 common isotope ratios that we've been discussing today, delta C13 on the x-axis and delta N15 on the y. Each point in this biplot is a set of carbon and nitrogen isotope values that represent an individual's diet during a certain time period. This time period is different for different metabolic tissues, which is related to their isotopic turnover rate. More on that later. Some tissues like blood plasma turn over in a few days and represent diet over a few days. Red blood cells turn over in weeks to months. And some tissues, such as feathers, claws, or hair, catalog the diet in their inert keratin structure as the tissue grows. Each diet is comprised of different major food sources, as you can see here, 1 through 3, each with their own isotopic signature in that location and time. For instance, these different food sources, which are displayed as primary colors, when combined, create different mixtures of colors representing different consumer diets. To estimate the diets of consumers, we must transform the foods so that they are comparable isotopically to our consumer tissues. To do that, we must add discrimination factors to our foods. These discrimination factors are those values I introduced earlier when talking about the predictable differences between the stable isotope values of consumers and their foods when describing food web structure. Once discrimination factors are applied, this transformation yields the stable isotope values of consumer tissues when feeding on each food source 100% of the time, and create this isotopic mixing space. This isotopic mixing space is used to estimate the proportions of each food that makes up our mixtures. To estimate dietary contributions using stable isotopes, we use stable isotope mixing models or mixture models that rely on the principles of mass balance and assume that the weighted sum of isotope values in dietary sources, plus these discrimination factors that we've added in, equals the isotope values of the consumer tissues. Since applying the wrong discrimination factors to your foods can alter your mixing space, as you saw here, and the proportional dietary contributions calculated by mixing models from this mixing space, we must be certain that these offsets are accurate. Many studies use discrimination factors from past studies that fed different animals different controlled diets. However, the best method is to conduct a controlled feeding experiment of your own, where your species of interest is fed the same or similar foods that they eat in the wild. Once their tissues fully equilibrate to the isotopic signatures of the food, you take the difference between the food and the equilibrated tissue and add it to the isotope values of your wild sources, as we did in this past slide. You got that? Well, if not, I'll demonstrate it here using a study that looked at discrimination factors in laboratory mice and applied those discrimination factors to bears. In these plots, we show how quickly carbon isotope values in 3 millimeter segments of mouse hair changed through time as mice were switched to 3 different diets of corn, meat, and C3 plants. At the start of the feeding trials, the mice had similar delta C13 values, but their hair quickly shifted towards the isotopic signatures of the diets they were eating. Mice fed corn became much more enriched in C13 because, as we've learned, corn is a C4 plant with a relatively high delta C13 value. In contrast, mice fed a mixture of C3 plants became more depleted in C13, reflecting the lower delta C13 values typical of C3 vegetation. And finally, delta C13 values for mice who fed on an all-meat diet also shifted rapidly like corn, but towards an intermediate carbon value because the meat diet reflected both C3 and C4 carbon sources. The key result of the study is that 3 millimeter segments of hair reached equilibrium with the experimental diets quickly, with estimated half-lives of about 6 days for corn, 7 days for C3 plants, and less than 5 days for meat. This showed that short hair segments can capture recent diet information and that experimentally derived discrimination factors can be used to better estimate the importance of corn, C3 plants, and meat in the diets of free-ranging monogastric omnivores. In the same study, we then applied the discrimination factors for mice to estimate the diets of brown bears in Slovenia that feed on supplemental corn across their entire human-dominated landscape, and black bears in Wisconsin that feed on hunter bait, which is primarily sweets loaded with high fructose corn syrup. We assumed turnover was the same for bears and mice since we found no relationship between carbon isotope turnover and mass. This allowed us to justify the use of food-specific discrimination factors as described for mice when estimating the proportional dietary contributions for bears that feed on the same food sources. After applying these diet-specific mouse discrimination factors to bear foods, we used a one isotope two-source model in each case. For Wisconsin black bears, males are in dark gray and females are in light gray, and subadults are the open circles, whereas adults are closed circles. We found that male subadults may have foraged for more hunter bait than other conspecifics in the population. Now, although understanding the importance of major food sources in bears' diets is important, like demonstrated with Wisconsin black bears, understanding how diet changes through time in response to some sort of factor is often more important. To demonstrate this, we also applied mouse discrimination factors to brown bears harvested or killed for management purposes in Slovenia in 2004, when the beechnut crop was in excellent condition, and in 2005, when the beechnut crop failed. As you can see here, some bears responded to the failed crop by feeding on more corn. Although stable isotope analysis of most tissues, including whole hair analysis, provides valuable dietary information over discrete time periods, and collectively analyzing many different tissues collected at the same time period can provide an understanding of diets over a broader time scale, their use in understanding time series over longer periods of time is often limited. In this study, we collected and analyzed tissues from bears in Yosemite National Park over the past century to evaluate the effectiveness of the management program in reducing the amount of human food available to bears on the landscape. So instead of measuring the amount of food on the ground through time, which is nearly impossible, we measured the amount of human food in bears through time. That's a proxy, then, for what was available to them over the past 100 years. To do this, we first had to build our mixing spaces for each time period in Yosemite's history, where we had tissues and knew the management actions of what was going on the landscape. We used the isotopic signature of bears that do not eat human food from our previous study as our natural food source. So these are already transformed by just running hair from bears that don't eat human food. They're already transformed into the isotopic signature of hair of bears that feed on 100% natural food and not human food. We also derived the signature of bear hair when bears eat 100% human food by sampling humans who also eat 100% human food. And we did this through time by sampling people's hair from the Trotter collection at the Smithsonian. We then calculated the dietary contributions of bears and learned that as management shifted in the park from reacting to food conditioned bears feeding in dumps to preventing bears from eating human food in the first place using proactive management, human food diet proportions also decreased through time, suggesting the park was in fact effective in reducing the amount of human food available to bears in the park. Okay, we finally made it. Now we can talk about our recent work. Despite the promise of using hair to reconstruct diets of animals through time, several challenges have limited its broader application in the fine-scale time series analyses that we hope to conduct. Most importantly, sampling animals through time or cutting up single hairs into small segments is difficult to prepare for laboratory analysis. In fact, if you want a graduate student to leave, have them do this. It's a real pain. Anyway, hairs segments might also be too small for reliable measurements using conventional IRMS and may be still too large to achieve the resolution required to detect the precise timing, direction, and magnitude of foraging events, such as diet switches and detecting individual level foraging events, like incidents when bears feed on human food. As a result, these hair segments often contain mixed isotopic signatures derived from multiple days or even weeks of foraging. To overcome these limitations, back in 2011, Jim Moran from Michigan State University developed a method for measuring delta C13 along individual hair strands using laser ablation isotope ratio mass spectrometry. For this approach, a laser ablated hair from a person and a horse at discrete 50 micron intervals. That's like 5 measurements for each millimeter of hair. This approach enabled high-resolution, time-resolved isotopic profiling along hair lengths corresponding to subdaily growth increments without physically having to section the hair. Boom. In this study, we applied the laser ablation method developed by Jim to measure delta C13 in the hair of 2 American black bears fed 3 different food sources: corn, white-tailed deer, and trail mix. So why trail mix, like this bear is eating here? Trail mix is now a premium hunter bait used to harvest bears in Maine and many other states and provinces in North America. The main objective of this preliminary study was to evaluate the potential of using laser ablation mass spectrometry for detecting diet shifts from isotopic profiles derived from bear hair. This work represents a key first step towards reconstructing fine-scale dietary timelines for free-ranging animals from hair samples collected from individuals with unknown foraging behavior like those in Yosemite that are involved in incidents. So a few years ago in the fall, we conducted 2 sets of feeding trials at the Wildlife Science Center, which is a private nonprofit organization located in Stacy, Minnesota. For each set of feeding trials, we fed each bear approximately 5 kilograms of food each day. For trial 1, bears fed on corn for 5 days, deer for 5 days, and then corn for another 4 days. For trial 2, bears fed on trail mix for 5 days, corn for 5 days, deer for 3 days, corn for another 3 days, and trail mix for the last 3 days. Corn was isotopically distinct from other food sources, and it was fed to bears prior to feeding them trail mix and deer. Each set of feeding trials ended on the day the animals were chemically immobilized and hair was sampled. When immobilized, before the trials began, we shaved each bear in the same location using portable grooming clippers, exposing patches of skin. We then treated each patch with Nair. After each trial, when immobilized, we pulled regrown guard hairs from each patch where hair had been growing during that time period using needle nose pliers. These short hairs are the focus of this project. Here, we've plotted delta C13 values that were measured along the length of each hair from tip to root, so the data are chronological. We assume that observed shifts in the isotopic value of the hair is related to a different treatment during each controlled feeding trial. We identified these shifts by looking for obvious changes in sign of the slope of the observed isotope values along the length of the hair. We matched peaks and troughs in each isotopic profile to known diet switch dates and used those intervals to estimate hair growth rate. We assumed that hair growth was constant within each diet treatment. For each diet treatment, we calculated hair growth rates as the length of the hair produced during the treatment divided by the duration of the treatment, so in millimeters per day. We then used treatment-specific scaling factors to convert the position of each laser ablation measurement along the length of the hair into time since the start of the trial, which provided the days on the x-axis displayed here. We found a clear isotopic response to each diet switch in the study. Although we assumed hair growth was constant when bears fed on each food source, growth rates varied among treatments for Ethel and Ricky. For both Ethel and Ricky, feeding on corn led to increases in delta C13 in hair, whereas delta C13 decreased when bears fed on deer and trail mix. As a next step, we will measure food-specific discrimination factors for black bears to be sure we can accurately estimate the delta C13 incorporation rates for each food source. By allowing the rate of isotopic change to vary among foods, future models could accurately detect a unique foraging event, like eating human food, and which food source best explains the observed delta C13 spike or decline. Finally, I'm happy to say that after 15 years of not working in Yosemite, we just got a grant from the Yosemite Conservancy to continue our isotope work. This time, we are in the process of linking isotopic profiles of Yosemite bears, which each contain 1 full year of diet data, to the movements of the same bears on the landscape to better understand bear habitat relationships and to build predictive models used to detect incidents when bears with and without unknown management status fed on human foods. Thank you so much for attending, and please feel free to reach out to me with questions. Take care and have a nice day.
Well, to those of you who joined us during Jack's presentation, the surprise is that he's actually not here. He had to pre-record his talk. Excuse me. He just texted a little while ago that he's not going to be able to join us for the Q&A, unfortunately. If people have questions for Jack directly, here's his email in the chat. Of course, you can reach us through the website or whatever. We have a couple of bear experts on here. If folks have questions, I can put Nate on the spot. His name is displaying prominently on my screen right now. Unfortunately, isotopes are not my specialty. I could talk plenty about sampling designs and whatnot, but not as much about. There he is. There's a true bear expert, folks. The premier biologist in Alaska, I will say. Definitely one of the premier locations.
Maybe
yeah,
the location.
If folks have questions, one of us might be able to field. We're happy to try. Otherwise, we can direct people to Jack. We can give lists of publications. Most of these will be published on our website soon. We are in the process of revamping our research project pages right now to a new format. So we'll be having updates for projects like the laser ablation work in the near future. And while people are still here, I'll put another plug in for upcoming talks. A month from now or so, we have another presentation. I'll stick with the plant theme in a sense. And most bears that Jack's talking about are mostly herbivores anyway, about the amazing adaptations that plants have for living in extreme environments, which would be, I think, very enlightening for a lot of us. A little bit off the wildlife path, but it's important to always keep in mind what our habitats can support. And then the month after that, in September, we have a project I'm helping to coordinate is a review on the bear rubbing behavior literature and trying to get a better handle on what that behavior actually represents. We do have one question here. Do the chemicals in hair affect the isotopes you see in the regrowth samples? Oh, in the nair. Having read a bit about this, I would think that's something they would have controlled for years ago when they first started using that technique. And there are actually publications I've seen recently that review, I think this was 2025, Nate, you might remember actually, where they contrasted different approaches for measuring variable diets via isotopes and how the different sampling techniques essentially pulled hair, the different tissue types, I believe, and then whether or not shaved versus plucked full hair, how those can affect your results. I honestly forget the outcomes. Like I said in the beginning, it is a much more nuanced analysis than what some of this appears to be. Jack just touched on some of the discrimination factors and whatnot. There's really is a lot to it. But I think some of the progress with these methods like the ablation is going to really catapult this forward, especially as they can bring in new isotopes and expand the range of the diet components we can measure. Another question. I was wondering if anyone from our team has explored doing diet analysis via DNA from scat samples. Coincidentally, yes, we're in the process of that. We're actually working with biologists in Wrangell St. Elias National Park and Preserve here in Alaska to look at bald eagle and brown bear, both scavenging and predation on salmon in the Copper River system. And we're working with biologists at Oregon State University to look at both the eagle and bear diets using metabarcoding is the plan. We're actually out collecting samples right now, and we're going to be contrasting stable isotopes via the hair with the metabarcoding to see what we're missing potentially in the diet. And then looking at much like what Jack talked about, in fact, look at the shifting of diets as the season progresses, how it might affect, say, moose calf predation by bears, at least what the relationships may be. So yes, absolutely, people look at DNA via metabarcoding and scat samples for diet analysis. Also, a pretty recent paper, actually, I'm not sure how recent it is, relatively recent, that looked at the potential for quantifying diet using metabar coding, because right now most of it is more presence absence. If an item is detected in the sample, versus trying to relate that to both the importance of the diet and then just at least a relative quantity of the different diet items, which would be a massive step forward, because obviously if something just pops up in the diet once or twice, that's not nearly as important. So it's be getting closer to how isotopes look at the assimilation of different diet items into tissue types. So definitely advancing on those fronts as well. Yeah, it's a good question. Thank you. Oh, we have some at Oregon State. Yeah, please, Alexa, reach out. I think you probably have my email address. If not, I'll put it in here in a second, where you can reach me through the webpage as well. And yes, always great to work with new folks, see what's going on. There are, as Nate also knows, I'm sure there are other projects here in some other national parks, Lake Clark, at least, maybe Kenai Fjords, but definitely Lake Clark, where there are also students looking at metabar coding stable isotopes for bear diets. We're been kicking around some ideas to try to standardize those methods and data collection and whatnot. So it is a pretty exciting time. Any other questions for us? I know I'm not the intended person to ask questions of, but yeah. So again, if people want to send this along to other colleagues, whatever, please refer them to our webpage, or well, the webpage for signing up for new presentations, and for the YouTube site for recorded presentations. Always interested in new ideas, new topics. Feel free to reach out to us. Even though Jack's not here, I was trying to end these by, of course, thanking everyone for tuning in today. And I'm not sure what our distribution was. Obviously, Utah, Georgia, saw a couple other states pop up. It's always nice to see the distribution. I know we got a few folks, McRae and others from Alaska up here as well, which is great. We really appreciate the support. And even though Jack wasn't here, I definitely want to extend a thank you to him. I've been excited to see him present some of his work for quite a while through this series. He's a very busy, very busy man. So it's great to see some of his work here. But definitely feel free to reach out and give him a hard time for his questions.